Today class, what we're going to do is we're going to be looking at flower structure and all the different intricate parts of a flower. We're going to use the ProScope and we're going to use our desktop computers to basically aid us in this process. It will give us a much better image. Also, you'll be able to capture the image, so when you see a good image that you like, what we can do is capture that image just by pressing our button here, and basically that will capture our picture. We can then print out our pictures, label all the parts later on, so that you can have evidence of what you've been working on. First thing we're going to do is we're going to grab one of our flowers and we're going to zoom in on it. And when we zoom in on it, what we're going to do is basically capture a picture. That's the picture I just captured. And what we're looking at here are the petals all the way around. And then in the center, our structures in here are basically, we can see one of the anthers, stamen, and the filaments, the anther and the filament. And down in here we can see the top part of the pistil, and so when we open this flower up, we'll be able to examine these particular parts a little bit better. Okay. The second picture that I want to take is I'm going to switch flowers. Let's grab this picture for a second. Okay. Whereas you had long filaments and male parts in our first flower, this is all condensed into a little bundle. So in the direct center, we have basically the female part surrounded by the male part. So a totally different structure. And we'll talk about strategies of why these structures might exist. Okay, class, when you're using the microscopes, you want to start out with your lowest power, basically your 50x lens. And that will give you an overview. It also will give you your largest field of vision. When you actually spot something that you want to look at, and when we actually dissect the flower, then we'll switch over to 100 power. That will give us a much closer image, smaller field of vision, but we'll be able to see in more detail. Lastly, when we're looking at pollen grains or internal structures, we'll probably want to switch one more time over to the 200 lens, which will give us our best magnification. The best way for you to capture images using this particular lab is we're going to use our microscope in the handheld fashion as opposed to putting it on a tripod. This will allow you more freedom of movement, and also you'll be able to manipulate it a little bit better with the flower. The images that I want you to capture is I want you to capture a side image so that you can see the petals and sepals. I want you to capture the base of the stem where the stem meets the flower. That will give us the sepals and what kind of arrangement it has there. Then I want you to look straight down inside the flower so that we can actually see the male and female reproductive structures. Then we can open up the flower by moving the petals to one side, or even removing a couple of the petals, and actually take a nice image of the male part and then take another image of the female part. Afterwards, when we dissect the flower, we'll actually take an image separately of the stigma, and then of the style, and then of the ovary, and then again of the anther, and then the filament. Later on, we can open up the ovary and look inside and look at the ovules and look at the carpels. So we'll have a complete picture of all of the parts of our plant. Science is discovery. We as human beings have a remarkable ability to question and ask why, and then the intuitive nature to try to find out those answers. By doing that, we create science. Science is a process by which we learn how things work. Students learn differently. Some can just sit back in a chair without a pencil in hand and take everything in. Some students have to write every word you say, including your bad jokes. Some students have to make a model of it, deal with it with their hands, draw a picture of it, physically see that. Investigations are the fun part. They don't have to listen to me, number one. Number two, they get to talk among each other. Handheld allows you flexibility. It allows you to move around. It allows the students to be a little bit more creative. What I do is I tell the students always start out with low power. Start out with a 50x lens. We can see more and we can see generally. And that way we can see, first of all, is it a complete flower? Does it have all of its parts? How is it arranged? And if it is not, Then we can look for another flower which is complete if we're looking for all the parts, okay? Then we can switch from that 50 lens to a 100 lens and actually look at, for example, the pistil. The pistil is the part of the flower that receives the pollen. Is it smooth? Is it irregular? Does it have little tiny cilia on it? Is it bare? Is it twisted? That's extremely important to know about, because when we look at pollen grain, they are all different shapes also. And therefore, the pollen grain that pistil has to receive in order to begin birth of another plant have to be matched. If they're not, then the pollen can be rejected. So, shape and size and intricacy is extremely important. And the only way I can describe that is I can't. I have to let them see it. Once a student has captured an image in the lab, there's several things that they can do with it. Number one, they can print it out, just as it is, and then they can label it by hand. If we have software, then we can do more. We can put it into the computer and use a digital imaging program to label it on the screen and then print it out. We can also do it to allow students to make presentations before the rest of the class. This lesson can be suitable just about any grade level. It depends upon the students you have in your class. You can definitely use it at elementary school level and do it just a simple lab. You can beef it up a bit as you go up through the elementary grades. Middle school works perfectly in any of the life science curriculum. And then in high school, you have life science. You also have some programs in biology, AP biology, different levels of biology, and also in environmental studies you can use it. I want them to be able to see a comparative anatomy. I want them to be able to look at one flower and see that the anthers and the stamens are arranged a certain way, and in what position they are near the female reproductive parts, okay, the pistil. And I want them to be able to look at other flowers and see that it's not all the same. And then I want them to be able to come up with reasons why. Okay? Why is this flower very widespread and the pollen mechanism is on the top? See, that's observation, and that's critical observation. And what we have to teach our students is to see and think critically.